Hello everyone and welcome to today's session. And today we'll be discussing the NCERT practice questions for the chapter Microbes in Human Welfare. This is the disclaimer. So what are the objectives for today's session? So today we will understand the basic concepts related to the chapter Microbes in Human Welfare by working through the NCERT practice questions. Okay, so these practice questions, they are based on some concepts. We will discuss these concepts by discussing these questions. Okay, so in this session, we'll be discussing question number 1 to question number 16, and these questions, they are related to the chapter Microbes in Human Welfare. Okay, let's begin with the first question. The first question is, which of these bacteria is responsible for converting milk into curd? Okay, so here we are examining how certain microbes play a role in our daily lives, especially in the food production. Okay, so let's have a look at the options. So option number A is Aspergillus niger, which is a fungus. Second option is Lactobacillus, which is bacteria. Option number C, Acidobacter aceti. And then last option is Streptococcus. What is the correct answer? Let's have a look at the answer. See, option number B, Lactobacillus is the correct answer. Now, the main bacteria involved in converting milk to curd is called Lactobacillus. It is a lactic acid bacteria. Okay. Now, these bacteria, they produce acid. So, lactic acid bacteria, they perform lactic acid fermentation. So, they produce acid. Okay, and this acid coagulate the proteins present in the milk. What is the protein present in the milk? That is casein. Okay, so these proteins, they clump together, they coagulate. Okay, and curd is formed. Now, lactic acid bacteria also enhances the nutritional quality of the food. So, it enhances the vitamin B12 content of the curd. So, eating curd is healthy. It is providing you essential nutrients, and then curd is very healthy for the digestive system also. Clear? One of the option was Aspergillus niger. So Aspergillus niger is a fungus. It is not a bacteria. It is used in industry to produce citric acid. Okay, then you have Acetobacter aceti. So this is another important microbe. So this bacteria is responsible for turning alcohol into acetic acid. Then you have Streptococcus. This is the bacteria, and this has a special medicinal use. These bacteria, they produce an enzyme called streptokinase, which is used as a clot buster. This enzyme is used to dissolve the blood clots in the patients and prevents the future heart attacks. Next question, who discovered penicillin? Okay, so penicillin is an antibiotic. So who was the person who discovered penicillin? So options are Ernest Chain, Howard Florey, Alexander Fleming, or Louis Pasteur. Okay, so what would be the correct answer? Okay, so this type of question help you to understand the historical breakthroughs in antibiotics. Okay, so let's have a look at the answer. See, option number C, Alexander Fleming is the correct answer. Let's have a look at the explanation. So penicillin was discovered by chance while Alexander Fleming was working on Staphylococci bacteria. Clear? So penicillin is obtained from the fungus Penicillium. Okay, full name is Penicillium notatum. Now Fleming, he noticed that the mold Penicillium notatum was inhibiting the growth of the bacteria. So then he isolated this antibiotic penicillin from penicillium. Now, Alexander Fleming, he received Nobel Prize in 1945 along with Chain and Florey. So Chain and Florey, they also received the Nobel Prize along with Fleming. So they were the scientists who realized the full potential of this antibiotic. Okay, clear? So this is an image showing you Alexander Fleming. Next question, which microorganism is used in the commercial production of citric acid? Clear? So microorganisms, they are like tiny factories. Some of the microorganisms, they can produce acid, alcohol, antibiotics and other useful substances. Okay, so which of the organisms produces citric acid? So what are the options? Option number is Aspergillus niger. Second is Acetobacter aceti. Option number C is Lactobacillus and option number D is Clostridium butylicum. So what will be the correct answer? Let's have a look at the answer. So option number A, Aspergillus niger is the correct answer. Okay, so Aspergillus niger is a fungus that is commercially used to produce citric acid. Aspergillus niger, you can think it of like a biological machine, okay, and it is producing this chemical, this compound, which is called citric acid. Now, citric acid produced in this way is widely used in food and pharmaceutical industries. It's a key ingredient in many products you use daily, okay? You are using that every day. And this shows how biotechnology connects to our daily lives. Okay, this is clear. Now let's move on to the next question. The next question is related to the biogas. Okay, so this question checks your understanding of which gas is produced in the largest amount during the process of the fermentation. Okay, this biogas is produced during the anaerobic, or it is produced by the anaerobic digestion of the bio waste. Okay, so question is, what is the main gas component in the biogas? So biogas is the mixture of the gases. What is the major component of biogas? So options are, option number A is carbon dioxide, option number B is hydrogen, option number C is methane, and option number D is nitrogen. What will be the correct answer? So let's have a look at the answer. See, option number C, methane is the correct answer. Okay, so methane is the primary constituent of the biogas. Okay, it is the major constituent of the biogas. Methane is flammable. Okay, that's why biogas is flammable. It is used in cooking. It is used for lighting. It is even used for electricity generation. So it is a biofuel. So methane typically makes up around 50 to 75% of its composition. Now this range can vary depending on the materials used and the efficiency of the process. So basically, biogas is produced by anaerobic bacteria such as methanobacterium, which is a methanogen. So methanogen, they produce methane. Okay, so biogas basically consists of methane, carbon dioxide, hydrogen, and a very small proportion of hydrogen sulfide as well. Next question, which bacteria is used as a biocontrol agent against butterfly caterpillars? Okay, so this question is about biocontrol agents. So biocontrol agents, they are living organisms used to control pests in a natural way instead of using the chemical pesticides. So we are reducing our dependency on the chemical fertilizers by using this biocontrol agent. Okay, so what are the options? Option number is lactobacillus, which is lactic acid bacteria. Okay, option number is Bacillus thuringiensis, which is a soil bacteria. Then option number C is rhizobium. Okay, rhizobium forms symbiotic association with the roots of the leguminous plant, and it can do nitrogen fixation as well. Okay, then option number D is azotobacter. So what will be the correct answer? Let's have a look at the answer. The option number B, Bacillus thuringiensis, is the correct answer. So Bacillus thuringiensis, this is a soil bacteria, and this bacteria produces Bt toxin. Okay, so this bacteria has a gene, Bt toxin gene, also known as cry genes. So this cry gene, it produces Bt toxin, also known as cry toxin. Okay, now when the bacteria produces this toxin, it produces this in inactive form. But when this toxin enters into insect gut, then due to the alkaline condition of the insect gut, this toxin gets activated and it creates holes in insect gut, and the insect finally it dies. Okay, so this Bacillus thuringiensis, also known as Bt bacteria, produce crystalline proteins, also known as cry proteins, which are toxic. Okay, now these Bt toxins, they do not harm animals or other animals because they are only activated in the alkaline conditions which are present in insect gut. Okay, so this makes it an effective tool to control pest caterpillars like those of the butterfly. So this Bt toxin is insect group specific. It targets specific insect. Okay, and there is no harm to other animals or other beneficial insect like bees. So bees are very important. Okay, they are very important for pollination. If there is no pollination, there will be no fruit and seed set. Clear? That's why Bt, Bacillus thuringiensis, this is a very useful biocontrol agent. Now, this gene has been introduced into crop plants such as cotton. So Bt cotton have been developed. So when the insect feed on this Bt cotton, this toxin goes into insect's gut. Okay, and then the holes are created in insect gut, then the insect will ultimately die. And in this image, you can see Bacillus thuringiensis. You can see here Bt toxin crystals are being produced by this bacteria, which is inactive form of this toxin. Okay, so Bacillus thuringiensis, this is a powerful example of how microbes can benefit humans, especially in agriculture. Okay, now let's move on to the next question. The next question is, in Swiss cheese, large holes are caused by which bacteria? Okay, so if you must have seen cheese, then there are holes in the cheese. How are these holes created? These holes are also known as eyes. Okay, so what are the options? Lactobacillus, Propionibacterium shermani, Streptococcus, or Acetobacter? What is the correct answer? Okay, so let's have a look at the answer. Say option number B, Propionibacterium shermani. This is the correct answer. Okay, so Propionibacterium shermani produces carbon dioxide as a byproduct of its metabolism during cheese fermentation. So this carbon dioxide is basically produced by the process of fermentation. What is fermentation? It is basically anaerobic respiration. Okay, means oxygen is not being utilized. Okay, so because of the production of carbon dioxide, these holes are created in Swiss cheese. Clear? And this carbon dioxide produced as a byproduct of the metabolism, as a byproduct of the process known as fermentation. Now, this carbon dioxide production leads directly to the characteristic large holes in the Swiss cheese. So the gas forms bubbles that are trapped in the cheese, creating those familiar eyes, which are also known as holes. It is a perfect example of how a simple organic biological process can have a visible effect. Okay, biological process is going on. This is fermentation. Okay, but you can see it as the holes are being produced by the evolution of the carbon dioxide. Next question, what does BOD stand for in sewage treatment? So they are just asking about the full form of BOD. Options are biological oxygen demand, second is biochemical oxygen demand. Option number C is basic oxygen demand, option number D is bacterial oxygen demand. What is the correct answer? Okay, so option number B is the correct answer, biological oxygen demand. Okay, BOD, biological oxygen demand, okay, is the amount of oxygen that bacteria need to decompose organic matter in the water over a specific period. This amount of oxygen used by the bacteria here while it is decomposing the organic matter in the water is known as biochemical oxygen demand. Okay, another term used for biochemical oxygen demand is biological oxygen demand. So these terms are used interchangeably. Here, option number A is also correct here. So this measurement is used as an indicator of the level of pollution in the water bodies. So higher the BOD, the more polluted the water is, because more oxygen is needed to break down the organic matter. Okay, next question, which yeast is commonly used in bread making? So microorganisms, okay, they can be used for the preparation of certain food as well, certain drinks also, certain alcoholic drinks as well. Okay, so these microorganisms, they are useful for humans. Here, what are the options? Option number A is Saccharomyces cerevisiae, which is yeast. Option number B, Penicillium notatum, which is fungus. Option number C, Aspergillus niger, which is again fungus, and Trichoderma, which is again a beneficial fungus. Okay. So what would be the correct answer? So let's have a look at the answer. Option number A, Saccharomyces cerevisiae, is the correct answer. So this is the scientific name of the yeast. Saccharomyces cerevisiae is also known as baker's yeast. So this is the specific species used in bread making, and it's famous for its ability to ferment the sugars and produce carbon dioxide, which help the dough rise. So that's why it is called baker's yeast also here, because it is used for bread making. Now what yeast does? Yeast, it feeds on the sugar present in the dough. Here it is basically fermenting the sugar present in the dough. Here, and which kind of fermentation is occurring? Alcoholic fermentation. In alcoholic fermentation, alcohol and carbon dioxide are produced. Clear? And this carbon dioxide, this causes the dough to rise, and it causes the puffier appearance of the dough. Now, this yeast is primary organism responsible for fermenting the sugars in the dough. Here you can see in this image is showing you dough fermentation, and you can see carbon dioxide is being produced by this process. Okay, so yeast basically carries out alcoholic fermentation. So you can see in this image, this dough consists of starch, Okay, now some of the yeast, they can produce amylase as well. So with the help of amylase, they can break this starch into simple sugar like glucose, and on this simple sugar, then yeast can act on this simple sugar and then thus producing carbon dioxide in this process. Okay, this is clear. Next question, which microorganism produces the immunosuppressive agent cyclosporin A? Immunosuppressive agent, they suppress the immune system. Immunosuppressive agents are used in the case of organ transplant. Okay, so you are lowering down the body's immune system by using this immunosuppressant. The options are option number A, Aspergillus niger, option number B, Trichoderma polysporum, option number C, Monascar purpureus, and option number D is Penicillium notatum. What is the correct answer? Let's have a look at the answer. See, option number B, Trichoderma polyspermum is the correct answer. So, Trichoderma polyspermum produces cyclosporin A. This is an immunosuppressant. Okay, cyclosporin A is an immunosuppressive agent used in organ transplant patients. So this means it helps prevent the body from rejecting a new organ, making transplant much more successful. So during transplant, what you are doing, you are inserting a foreign tissue in the patient's body. Okay, so body recognizes this organ, transplanted organ as foreign. Immune response will be generated and it will target this new organ. Okay, it will target the tissues of these organ, and the organ rejection can occur. So to prevent organ rejection, immunosuppressants are given to the patient. Clear? So they are very useful in organ transplant patient. Next question, which of these is the biofertilizer? So basically biofertilizer, they are living organisms that help enrich the soil by fixing nutrients, especially nitrogen, making them available to the plants. What are the options? Rhizobium, Penicillium, Lactobacillus, or Saccharomyces? What is the correct answer? Option number A, Rhizobium is the correct answer. So Rhizobium, this forms symbiotic association with the roots of the leguminous plant. It is a type of bacteria, symbiotic bacteria. Rhizobium can carry out atmospheric nitrogen fixation. Okay. Rhizobium forms symbiotic relationship with the leguminous plant. In symbiotic association, both the partners are benefited. Clear? Now, this bacteria fixes atmospheric nitrogen into usable form for the plants, making it as a key biofertilizer. Okay, you can see in this image, this is an image showing you rhizobium. You can see rhizobium, they are living in the root nodules of the leguminous plant. So they form root nodules here and they live inside this root nodules. This is a symbiotic association. Okay, this is clear. Now let's move on to the next question. Next is, what is activated sludge in the sewage treatment? Here, so sewage treatment is very important. Prior to disposing this sewage into the rivers, the sewage water is treated. So there are different stages: primary treatment and secondary treatment. Okay, so they are asking about what is activated sludge in the context of sewage treatment. Now options are raw sewage, bacterial flocks, primary filtrate, and untreated waste. What is the correct answer? Let's have a look at the answer. The option number B, bacterial flocks, is the correct answer. Now, activated sludge consists of bacterial flocks. So, flocks or clumps. So, these are clusters of bacteria that stick together, along with some fungal filament also, and they form visible clumps in the treatment tanks. And these flocks, they are the major agents responsible for breaking down the organic matter in the sewage. These flocks are made up of primarily of aerobic bacteria along with a fungal filament. Now, the bacteria need oxygen to survive and work efficiently, while the fungal filament help hold the flock together, making them more stable and effective. Here, now these bacterial flocks are used to biologically treat sewage by consuming organic pollutant during the secondary treatment process. This means the microbes feed on the water, breaking it down into simpler, less harmful substances, which help purify the water before it is released back into the environment. Clear? Let's move on to the next question. The next question, which enzyme produced by bacteria is used as the clot buster? Okay. Again, you can see microorganisms, they are being used in human welfare. Microorganisms, they are being used in medicine as well. They are used for treating heart attacks also, or blocked blood vessels. So what are the options here? Lipase, actinase, streptokinase, or protease? What is the correct answer? Okay, let's have a look at the answer. Option number C, streptokinase, is the correct answer. So streptokinase is an enzyme produced by bacterium Streptococcus. Okay. Now, this enzyme is used to break down blood clots in the blood vessels. So this makes streptokinase a valuable agent in treating conditions like myocardial infarctions or heart attack. Now, by dissolving the clot quickly, it helps save heart muscles and can be life-saving. Yeah, so this is very important, streptokinase. So you can see microorganisms, they are being used in human welfare, they are being used in the medicine area also. Okay. The next question, which organism produce statins? Okay. So microorganisms, they are producing a lot of compounds, and these compounds have been isolated by humans and they're using it for the human welfare. Okay, so microorganisms, they are acting like tiny bodies. They are making chemicals and they are helping to treat diseases and improve health. So basically, these chemicals are helping to treat diseases and improve human health. Okay, so this question is testing your knowledge, okay, of microbes producing these chemicals. Okay, what are the options? Trichoderma, Monaskos purpureus. Option number C is Aspergillus niger. Option number D is Penicillium. What is the correct answer? The correct answer is option number B, Monaskos purpureus. Now, the yeast Monaskos purpureus is known for producing statins. Okay, statins are used to lower the blood cholesterol level. Now, this is important because high cholesterol can lead to heart diseases. So, these medicines play a big role in keeping people healthy. Let's move on to the next question. The next question is, which part of cattle is methanobacterium commonly found? So methanobacterium is basically comes under the category of methanogens. This methanogen, they produce methane gas. They are aerobic bacteria. And whenever they feed on, you can say, bio waste, okay, organic matter, okay, and this organic matter, if it is rich in cellulose, okay, they act on this organic matter and they produce methane. Clear? So where are these organisms commonly found? In cattle. The options are liver, rumen, intestine, or stomach. What is the correct answer? So let's have a look at the answer. See, option number B, rumen is the correct answer. So methanobacterium is a type of bacteria. It is a type of aerobic bacteria commonly found in the rumen of the cattle. Okay, rumen, it is a part of stomach in the cattle, and these are anaerobic bacteria. They do not require oxygen, so they carry out fermentation. Okay, and in the process they produce methane. Okay, which is a major component of biogas. Now, next question: which microorganism is used in the production of acetic acid? Again, a very straightforward question. Here options: Acetobacter, Aceti, Aspergillus niger, Lactobacillus, or Clostridium. What is the correct answer? Okay, let's have a look at the answer. See, option number A is the correct answer, Acetobacter aceti. This is the correct answer. So, Acetobacter aceti, this is a species of bacteria. Means bacteria are what? They are prokaryote. They are single-celled organism. They are unicellular. Okay, but these bacteria, they have a very powerful ability. They can synthesize these compounds. Clear? So, acetic acid is being produced by Acetobacter aceti. Now, Acetobacter aceti can oxidize ethanol into acetic acid. In simple term, it takes alcohol and with the help of oxygen transform it into vinegar's main component. So basically it is converting alcohol into acetic acid, here, which is a component of the vinegar. Now this process is fundamental in vinegar production. Without Acetobacter aceti, we wouldn't have acetic acid and therefore no vinegar. So this shows how essential microorganisms are in making everyday products. Next question, which of these is a mycorrhizal fungus? So the term mycorrhiza, what does this mean? So myco means fungus, rhiza means roots. So these mycorrhizal fungus, they are special type of fungus that form a partnership with the plant roots. Okay, so here both the partners are benefited in symbiotic association. This is a kind of symbiotic association. Fungus is also benefited, plant is also benefited. Okay, so plant, okay, it provides food to the fungus, it is providing shelter to the fungus, and the fungus is helping the plant to absorb nutrients and water from the soil. Okay, it is solubilizing the phosphorus and it is making, you can say, it is transferring the phosphorus to the plants. Okay, so plant cannot directly absorb phosphorus from the soil, so it converts the phosphorus into a form that can be absorbed by the plants. Okay, so you have to tell which of these are mycorrhizal fungus. This is a mycorrhizal fungus. Options are Penicillium, Aspergillus, Glomus, or Trichoderma. What is the correct answer? Let's have a look at the answer. See, option number C, Glomus is the correct answer. So this is a genus, okay, of arbuscular mycorrhizal fungus. Okay, this means Glomus belongs to a group of fungi that form special structures inside the plant roots, helping them absorb nutrients. Okay, these fungi form symbiotic relationship with the plant roots. Now, symbiosis means both the fungus and the plant benefit from this partnership. Okay. Now, glomus fungi actually penetrate the root cells and help the plant absorb nutrients from the soil. Okay, clear? And the fungus is getting the food from the plant because plant can photosynthesize. It is producing the organic compounds. Okay, so with this, we have discussed some of the NCERT practice questions related to the topic microbes in human welfare. Okay, I hope your concepts are clear now. Okay, so with this, we will end this session. I hope this session was quite knowledgeable for you. We'll meet you in the next session. Thank you very much for listening.